Thank you, everyone. I first want to thank Dr. Chambers and the Foundation to give me the opportunity to speak. And this afternoon, I will be discussing DADA2 and the role of the endothelium. So, as you all know, one of the key clinical features in DADA2 is vasculopathy of the small and the medium-sized arteries. And these manifestations range from cutaneous vasculitis with livedo racemosa, polyarteritis nodosa, and a nose phenomenon, but also neurological involvement with early onset life-threatening strokes that are partially responsible for the mortality rate we observe in DADA2, but also other end organ involvement with liver, kidney, and gastrointestinal problems, with hepatic and renal aneurysms and abdominal pain. And these patients are mostly helped when they are treated with TNF-alpha inhibitors. However, over the past year, the clinical phenotype has expanded, including bone marrow failure with bone marrow with neutropenia and thrombocytopenia, and these patients are often treated with hematopoietic stem cell transplantations. Now, the role endothelium and DADA2 has a story that's already started in the beginning, in one of the first publications by Zhu and colleagues, where they observed intracranial hemorrhage when zebrafish were injected with CEC or1 morpholidols that blocked ADA2 expression. And this pointed either problems with vessel development or endothelial cell integrity. However, the vessel morphology seemed fine, so this pointed more to a problem with endothelial integrity. And moreover, when they looked at biopsies of DADA2 patients, they observed endothelial activation as well as inflammation, and moreover, co-culture experiments of endothelial cells with monocytes derived from patients showed that there is a problem with the endothelial monolayer. So now the role of the endothelium and the expression of ADA2 by the endothelium is a topic of debate. So based on what they have observed with the intracranial bleeding in the zebrafish, Zhu et al. Looked at ADA2 expression both on qPCR level in different endothelial cells, as well as intracellular ADA2 expression. And they could observe only low levels of ADA2 and no intracellular ADA2 expression in the cell. And this implicated additional cells in the vasculopathy observed and a more passive role for the endothelial cells. However, which has already been discussed this morning, there is another paper by Dawani et al., and they show that there is mRNA expression of ADA2 in different endothelial cells, and moreover, they also showed secretion of ADA2 in UVEX. And they proposed, as what has been discussed already this morning, that a lack of endothelial ADA2 triggers a type 1 interferon response. Now, when we take a step back and just see two elements that could support a more active role of the endothelium in DADA2, I have three elements that could support that. So first of all, the stem cell niche. Endothelial cells are present in the stem cell niche, and they produce very important factors, angiopoietin, CXCL12, and CCSF, which are all very important to maintain hematopoietic stem cells. They have also shown that when sinusoidal endothelial cells are really important for hematopoietic recovery after myoablation. And in vitro experiments also show that endothelial cells play a role in the proliferation of hematopoietic stem cells. So it's a very important part of the stem cell niche. Next to that, they also play an important role in the immune response. First of all, because of their position, they're the first cells where pathogens need to interact with, and therefore they express different pathogen recognition receptors, including TLRs. But also they're the first cell that immune cells need to interact with. Moreover, besides their position, they also have immunomodulatory properties by both producing pro-inflammatory and anti-inflammatory cytokines and chemokines. And lastly, vascular pathology that is observed after hematopoietic stem cell, for example, in the form of nodular regenerative hyperplasia of the liver, and this may suggest an incomplete rescue of the DADA2 pathology after hematopoietic stem cell transplantation. So to have a look whether endothelial cells express ADA2, we looked at an array of endothelial cells that originate from different organs that are affected in DADA2 pathology, and we performed the screening in 3 ways. So first we looked at ADA2 mRNA expression by qPCR. Next we looked at ADA2 intracellular expression in whole cell lysates by Western blot, but also via intracellular staining via flow cytometry. And lastly, we also looked at ADA2 protein secretion. So first of all, we had a look at ADA2 mRNA expression, and for that, we included as a negative control HEK cells, and as positive controls, U937 and THP1s were used, both monocytic cell lines, as well as primary monocytes derived from a healthy control. And as you can see here, we observed variable expression across the different primary human endothelial lineage cells, but I want to point out that we observed some similar levels of expression between the coronary artery and brain endothelial cells when we compare them to monocytic cell line THP1s. And we had a look at lung endothelial cell ADA2 mRNA expression. It is around the same level of the U937s, another monocytic cell line. So next we looked at ADA2 expression inside the cell, first via Western blot by looking in whole cell lysates, and we used the same positive and negative control. So again, the HEKs were used as a negative control, and U937s and monocytes were used as a positive control. And as you can see here, we were not able to detect any ADA2 protein inside these cells. So we moved over to another technique to have a look at intracellular ADA2 expression by flow cytometry. So we first permeabilize the endothelial cells and then perform intracellular staining with the same antibody we use for Western blot and then a fluorescent secondary. And as you can see here, there is clearly a shift in ADA2 to the right in the U937s, which we used as a positive control, which is absent in the HEK cells. But we observed also a slight shift in peak in the coronary artery, the brain endothelial cells, but also in blood outgrowth endothelial cells. So lastly, we also had a look at ADA2 secretion in all of these different primary human endothelial lineage cells, and therefore we see the 200,000 cells per well, and we incubated it for 48 hours in serum-free medium. Then we concentrated the supernatant and we load 40 microliters of the supernatant on a gel. And again, the same controls are included. And as you can see, we again observe a variation of secretion of the ADA2 protein across the different endothelial cells with remarkable secretion in the coronary artery, the brain, as well as the blood-load endothelial cells, which correspond a bit with the previous data. So now that we have looked into ADA2 expression and secretion, we could ask ourselves what could be the role of endothelium in the DADA2 pathology. It could be that the endothelial cells might aid together with blood cells and tissue macrophages in controlling excessive inflammation in healthy individuals, and that when ADA2 is not present, the excessive inflammation occurs. Moreover, there's also a paper where they performed co-culture experiments with parasites, UVEX, and macrophages, and they showed that when ADA2 is knocked down, this inhibited neoangiogenesis, while when they add ADA2, this promoted neoangiogenesis, and this might explain the alveolar hemorrhage, myocardial infarction, and arterial hypertension in young age in DADA2. And lastly, the loss of ADA2 in these endothelial cells might have an effect on the functionality of the endothelium, especially in organs where the flow of immune cells is not as prevalent, where the ADA2 of the endothelial cells might play a role. So in conclusion, we screened an array of primary human endothelial lineage cells that originate from different organs affected in ADA2 deficiency. And we have shown that endothelial cells express ADA2 or mRNA, although at lower levels compared to primary monocytes. Some were comparable levels to the U937s and THP1s, which are often used as models in NADA2 studies. Intracellular ADA2 expression was also observed in coronary artery and brain microvascular endothelial cells. And we also observed a variable secretion of the ADA2 protein by these different cells. So these findings open up new avenues in the exploration of understanding the role of an endothelium in NADA2 pathology, especially in organs where immune cells, where the flow of immune cells is low. I'd like to thank you for your attention.